Recombinant DNA molecule can be created normally by cutting the vector DNA and source DNA respectively with, so the answer will be same enzyme, option 1. So this is a question which is normally asked as in the format of whether the enzyme will be same or different, or combination of two different or three different enzymes. But in this case, they have given names of enzymes and asked you. The highlight is you have to treat both vector DNA and source DNA with the same restriction enzyme. So let's understand this question using the example which is given in NCERT textbook. So the recognition sequence GAA TTC. This is the recognition sequence of restriction endonuclease ECO R1. So it is going to break the bond between G and A. The complementary strand will be C TT AA G. Now this is vector DNA. Vector DNA. Now to ligate, the source DNA should have identical sequence of EcoR1. So it will also have GAA TTC. The complementary strand will have CTT AAG. Now in red color, I'm going to show you the side at which EcoR1 will attack and break the bond. So over here, and you need to treat, see the question is asking you normally by cutting vector DNA, source DNA respectively with the same restriction enzyme. So I will have to treat the source DNA also with EcoR1. So the after restriction endonuclease digestion, it will look something like this. Since this bond is broken, this strand will open up. So you will have only G, then C, and T, T, A, A. So vector DNA will have this overhang of TTAA. This is vector. What about source DNA? In case of source DNA, this strand will also open up. So you will have ATTC on one strand and only G on the other strand. This is source. Now these are known as sticky ends. Sticky ends. Because they have overhangs. Overhangs of AATT overhangs. Okay, so AATT, they will naturally complement with this area and they are going to hydrogen bond. So base pairing will occur between A and T, this one. Okay, then this is going to pair with the second T. Now this T will pair with the adenine and the last T is going to pair with this adenine. So now you will have a recombinant molecule which will look something like this. AATTC 1G and With the green color, I will show you the vector. So G, and then this will have C, T, T, A, A. So now we have a fusion of vector DNA and source DNA. And this fusion is fused DNA is called recombinant DNA because now it has the component of vector DNA as well as source DNA. It is new combination. Therefore, it is called recombinant DNA. So the highlight is both vector and source DNA should be treated with the same restriction endonuclease because it is going to recognize the same recognition site and cut it, and similar ends will be produced that can complement with each other, and then DNA ligase can permanently seal the two molecules together. So that's all about this question. Thank you so much for watching my video.